गुड मॉर्निंग क्लास। आज हम लोग जो है 4th यूनिट से स्टार्ट करेंगे। 4th यूनिट आपकी जो है वो बायो टेक्नोलॉजी वाली यूनिट है। उसमें जो फर्स्ट चैप्टर है दैट इज़ अबाउट द बायोटेक्नोलॉजी प्रिंसिपल्स एंड प्रोसेसेस। स्टूडेंट, इस चैप्टर में बायोटेक्नोलॉजी कौन से प्रिंसिपल पे बेस है? ये टेक्नोलॉजी बायोटेक्नोलॉजी मीन्स द टेक्नोलॉजी in which we are going to use the living organism. We are going to manipulate the genome or the DNA of the living organism. तो इसमें कौन से प्रिंसिपल्स इन्वॉल्व हैं और किस प्रोसेस से ये टेक्नोलॉजी होती है, यानी ये टेक्नोलॉजी करने के लिए हमें कौन कौन से प्रोसेस और प्रिंसिपल्स की रिक्वायरमेंट है। सो टुडे वी आर गोइंग टू स्टडी द बायोटेक्नोलॉजी, the principle and the processes. In this chapter, first we are going to know the definition of the biotechnology. Actually, biotechnology is the use of the living system and organism to develop or make the very useful products, or we can say any technological application that uses biological system, living organism or their derivatives thereof to make or modify the products or processes for the specific use. There is a federation of biotechnology named the European Federation of Biotechnology, EFB. This federation defined the biotechnology as the integration of the natural science and organism, the cells, their parts thereof, and the molecular analogues for the products and the services. Means this is the science in which we integrated the different organism, means like they're using their cells, their parts or their molecular analogues also for the products and services for the human being. Ancient time, we know that there are very oldest biotechnological examples. For example, making bread or making curd with the help of the microorganisms. A very oldest forms of the biotechnology means the manipulation of the biological organism or their things or products for the human being. Biotechnology. The few applications of the biotechnologies are the fermentation. We know that from ancient times we used the different fruits for the fermentation, for the production of wine and other alcoholic beverages, is also known as the biotechnological techniques. Two important techniques which enable the development of the modern biotechnology. There are two important techniques. One technique is the alteration of the chemistry or the molecular basis of the DNA and RNA to introduce into the host organism to change their phenotype of the host cell. Means it is known as the genetic engineering. So genetic engineering is the branch in which we are going to alter the chemistry of the DNA and RNA for the change of their phenotypic character. And second very important technologies things which is used in the development of the modern biotechnology is the maintenance of sterile ambience to enable the growth of the desired microbes or eukaryotic cells in a very large quantity for the manufacturing of the biotechnological products like the vaccine. So nowadays this genetic engineering is used in the production of vaccine, enzymes, beverages, drugs, and this branch is known as the chemical engineering. In the asexual reproductions, we know that the organism preserves its genetic information. And in the sexual mode of reproduction, which is also known as the hybridization, it leads to variation also, and it includes the so many undesirable genes with the desirable gene also. So genetic engineering isolate and introduce only one or the set of the desirable genes without introducing the undesirable genes into a target organism. And the techniques of the genetic engineering is the creation of the recombinant DNA. It is known as the rDNA technology. By the using of the gene with the help of the clone formation of the clones and the use of the gene cloning and gene transfer into the host cell. So recombinant DNA, or we can say the rDNA or alien DNA, it cannot multiply itself until integrated into the host genome. So when inherited into the host DNA, the ability to replicate due to the origin of replication, which is found inside the host DNA, it initiates the process of the replication. The alien DNA, or we can say the link, it is going to be linked with the host DNA for the process of replication and multiplication itself along with the host DNA. Actually, this process is known as the DNA cloning, in making the so many copies of the desirable DNA. In the next step, we have to transfer the gene, means the gene transfer to the host that is required, the vector. Without the help of the vector, we didn't transfer the gene into any host cell. So commonly used vectors, very common used vector is known as the plasmid. We know what is the plasmid. Plasmid is the small circular double-stranded self-replicating extra chromosomal material that is present in the cytoplasm of the bacterial cell. And in the first recombinant DNA, actually it is constructed by the Stanley Cohen and the Harvard Boyer in 1972. They have linked the gene encoding for the antibiotic resistant gene with the plasmid of the Salmonella typhi. Salmonella typhi is a bacterium, and they use the plasmid of this bacterial cell. Isolation of the desirable gene in the next step, the antibiotic resistant gene. After isolating this desirable genes from the host cell, we have to cut it into very small pieces of the DNA from a plasmid that is responsible for the antibiotic resistant, which involve the molecular seizure also with the help of, and this molecular seizure is also known as the restriction enzymes responsible to cut down the desirable DNA into small fragments. And this desirable gene, or we can see it is the alien gene, now it is going to be linked with the plasmid or the vector to transfer into the host cell. So with the help of the DNA ligase, as we know that a DNA ligase is used for the linking of the DNA or the joining of the DNA fragments. So with the help of the DNA ligase, the DNA molecules join their ends, and the new recombination, this circular autonomously replicating DNA that is created in the vitro chamber. So it is known as the recombinant DNA. It is a recombinant DNA made by human being, made by scientists also. And this recombinant DNA, now it is going to transfer into the E. coli. Actually, E. coli or the Escherichia coli, a very commonly used bacteria cell for the host for the replication. This DNA now replicate by using the host DNA polymerase enzyme, and after going multiple very steps, the multiple copies of the antibiotic resistant gene is going to form, and the cloning of E. coli will become also the antibiotic resistant. So the three basic steps that is used in the genetically modified organism. Number one is the identification of DNA. First we have to identify the DNA with the desirable genes. What we are going to use means what are the desirable genes present in the any microorganism. And identification, then introduction of the identified DNA into the host also, and maintenance of the introduced DNA in the host, and transfer of this DNA into its progeny. It is going to be maintained in the laboratory. So these are the basic steps that is involved in the genetic modifying organisms. In this picture, these are the steps of to genetically modified organism. Step 1. Step 1, you can see insulin plasmid is there, insulin gene is there, bacterial. In the bacterial cells means a small piece of the DNA, and it is cut down in the second step with the endonuclease enzyme, and after the ligazing of this into the circular DNA, in the next step, in the third step, human insulin DNA responsible for development of the human insulin, and in the fourth step it is going to be introduced into the plasmid cells, inherited into the new bacterium cells, and after using the bacterial replication process or DNA polymerase enzymes, so many copies of the insulin is formed in the last step. So bacterium divides and begins the producing the insulin in vitro chamber. These are the steps. First is the isolating genomic DNA. Second step, fragmenting it with the help of the endonuclease enzyme. Then screening, we have to identify the desirable DNA fragments and inserting it into the DNA of the vector. And after liging formation of the recombinant DNA, it have to introduce into the host cell, and with the help of the host DNA, it culturing into the cells, and it provides so many copies of this desirable genes. So transformation of the host cell. These are the basic steps that involve in the process. You can see here isolating genomic DNA from the donor, any donor, and the fragmenting this DNA using the molecular scissor. Then screening that fragments for desirable genes, whether it is present or not. Then inserting the fragments with the desirable genes into the cloning vector with the help of DNA ligase. Then introducing the recombinant vector into a competent host for making so many copies, and introduce into the culturing media to obtain multiple copies or clones of the desirable DNA fragments. By using these copies, to transform the suitable host cell so as to express the desirable genes. These are the basic steps that involve in the process of the biotechnology. Here you can see the molecular biology is interlinked with the biotechnology, microbiology, biochemistry, immunology, genetics, chemical engineering, and cell biology. These branches of the life science is introduced or combined with the biotechnological field and provide us which things? Provide a very desirable characteristics or products in the crops, in the agriculture fields, in the drugs, formation of the drugs, in the formation of so many vaccines, and many diagnostic techniques is also used, and it is also useful in the livestock. So thank you. This is the introductory session. Thank you and have a nice day.